Good evening, ladies and gentlemen. We cordially welcome you to the International Technical Webinar on Advancements in Biotechnology, presented by the Department of Biotechnology, St. Joseph's College of Engineering. Glad to have a wide range of viewers for our initiative. Now, we will start this session with a Tamil Thai Vartha.
சீராரும் பதனமென திகழ்வரத கண்டமெனில் தென்கணம் பதிச்சிறந்த திராவிடனை திருநாளும் தக்க சிறு திரைநுதலும் தரித்தனரும் திலகமுமே அத்திலக வாசனை போல் அனைத்துலகும் இன்பமுற எத்திசையும் புகழ் வழங்க இருந்த பெரும் தமிழகமே தமிழகமே உன் சீரிழமை திறம்பியந்த செயலறந்து வாழ்த்ததுமே வாழ்த்ததுமே வாழ்த்ததுமே.
Thank you, everyone. Now we will broadcast a small video clipping about our college, St. Joseph's College of Engineering. Now I warmly welcome our organizer, Mrs. Yorani, Assistant Professor, Department of Biotechnology in SJC, to welcome a speech.
Thank you, Sangeeta. Good evening to all. It is my privilege and it gives me an immense pleasure on behalf of Department of Biotechnology, St. Joseph's College of Engineering, to welcome all the faculties, research scholars, and students from various institutions from different countries for a 3-day international technical webinar on advancements in biotechnology. My warm greetings and special welcome to the speaker of the Day 1 webinar, Dr. REMYA MOHANRAJ. I welcome you, ma'am. And this webinar is designed to fill the gap of knowledge sharing that has been laid down by COVID-19. I once again welcome all the participants who has joined and waiting to grab the opportunity to explore the recent advancement and development in the field of biotechnology. Now, I request Dr. G. Srikumar, Professor and Head of the Department of Biotechnology, to deliver the inaugural address.
Good evening, everybody. I'm extremely delighted to know that the Department of Biotechnology is holding an international technical virtual lecture series on advancements in biotechnology, which is going to be held for 3 days between 22nd and 24th of this month. In this moment, I wish to bring some of the highlights about our college and the department. St. Joseph College of Engineering is one of the top colleges in India with the 160th position as per current NIRF ranking. And it has got 26 years of high reputation in educational society with the NAC A plus grade of accreditation. It has having 10 UG courses and 7 PG courses with affiliation from Anna University, Chennai. Our Department of Biotechnology is 1.8 decades old, having BTech and MTech biotechnology programs. Or we have bagged 139 university ranks with 10 golds and 7 silver medals, and achieving always 100% pass percentage every year. The department has got high-end, sophisticated laboratory facilities and fetched a grant research recognition status from Anna University. To do PhD program are having more than 1.1 crores of government funding to conduct research. In my view, nothing is more important in the present world than striving for good against COVID, with the honest collaboration and among individuals and peoples, nations, etc. Both students and faculty tend to lose their interest in subject due to continuous stress caused by pandemic outbreak. This technical lecture is like this will going to give us an affable environment for the academicians and research scholars to freely exchange their views with the actual practicing personalities. By keeping all these things in mind, the Department of Biotechnology has decided to organize virtual technical lecture series to keep our minds abreast to ongoing global research trends. In this regard, we gathered a global family of people who replenish our scientific resources and bring new visions to shape up the future strategy of biotech research. The spectrum of intellectuals for this 3-day international webinar series are Dr. MOHANRAJ. She's a faculty, Houston Community College, Texas, USA. Dr. Vijay Bhuminathan, a research scientist, University of Florida, USA. And Dr. Giridhar Shekhar, research associate, Albert Einstein College of Medicine, New York. And Dr. Sangamitra Saha, a lecturer, University of Houston, Texas, USA. I also appreciate the overwhelming interest shown by the registered participants. There are 1,102 participants, including 238 faculty members and 864 students, research scholars. The participants are from 9 different countries such as Singapore, Malaysia, Nigeria, Zambia, Ethiopia, Indonesia, UK, China, and Uganda, etc. There are 26 Indian states are involved in this participation, from Jammu Kashmir to Tamil Nadu and Punjab to West Bengal, all over the stretch of India. With this proud note, on behalf of the patrons of St. Joseph College of Engineering, I welcome the invited speakers and all the participants and inaugurate this virtual international lecture series of advancements in biotechnology. This is going to be definitely an actual excellent opportunity to all the delegates to exchange information on the latest biotech developments. I take this opportunity to convey my warm felicitations to the team of staffs and student organizers and participants, and extend my best wishes for the success of the lecture series. All the best. Thank you. Over to Ms. Sangeeta. Thank you.
Thank you, sir. Thank you for your warm welcome. With no further delay, we would welcome a star of the show, great speaker, Dr. REMYA MOHANRAJ. Dr. REMYA MOHANRAJ has completed her Bachelor of Science, majorly in Botany, postgraduate diploma in Bioinformatics, and Master of Science in Plant Science, all at Bharatiyar University, Coimbatore. Then she went on further pursuing Master of Technology in Biotechnology at Satyabama University. She attained her milestone by getting a PhD in plant tissue culture and phytochemistry in Bharatiyar University, Coimbatore. Her achievements are non-terminating. She has 21 international publications, presented 23 papers in conferences, attended 6 conferences, organized 2 national level conferences, attained 12 faculty development programs and training programs, and received 6 awards. She has a great honor as a reviewer and editorial board member for many specific scientific journals. She had a research grant fund of rupees 26 lakhs 48,290. Currently, she is working as a faculty of biology in Houston Community College System, Houston, Texas, USA. Today, she will be enlightening us a knowledge on research on medicinal plants and evolving trends. It's a honor to welcome you, ma'am. I request Dr. REMYA MOHANRAJ to take over the session. Thank you.
Thank you for the wonderful introduction, Sangeeta. I hope you can all hear me. And I would like to start my session without much delay. And thank you and welcome you all for today's presentation. And my presentation for today is going to be on evolving trends and research on medicinal plants. I would like to start this presentation with an introduction of medicinal plants. And for centuries, if you see, mankind has been utilizing medicinal plants for various purposes. If man had an ailment long ago when he was a hunter-gatherer or when he was an agronomer, he used to always rely upon medicinal plants. If you look at history, the references to medicinal plants has been found on a Sumerian slab, which was approximately 5,000 years old. The Vedas, the Indian holy book, they have a reference to medicinal plants as well. Homer's Iliad and the Odysseys, which date back to 800 BC, even they have a reference to medicinal plants. Up to the 18th century, medicinal plants were mainly used as in the form of crude extracts, powders, or decoctions. The breakthrough came in 19th century when they started using isolated compounds from medicinal plants. And it is during the 19th century that the structure of many of the medicinal compounds were also elucidated. Some of the compounds like strychnine, quinine, caffeine, atropine were all isolated during the 19th century. Today, according to World Health Organization, about 25% of the modern medicinal plants, or 25% of the modern medicines are basically from medicinal plants, and about 3.5 to 4 billion people depend on traditional medicine for their primary healthcare needs. What makes plants this important? What confers them with this unique medicinal properties? These are called the phytochemicals. Phytochemicals are simply put nothing but plant-based chemicals, and these are the chemicals that give the plants the medicinal properties that they have. In plants, the phytochemicals have 2 roles to perform. One is to give the plants the basic functionalities like growth and development. Another purpose of phytochemicals is to help the plant in defense mechanisms, to help them protect themselves from pathogens that attack them, to help plants protect themselves from herbivores that eat them, to help plants protect themselves from biotic and abiotic stress that they might undergo. So when phytochemicals perform this function, the extra function of giving them the protection, you call them secondary metabolites. And it has been proven that these secondary metabolites have a lot of medicinal properties. And you could classify them mainly as polyphenols, terpenoids, alkaloids, phytosterols, and organosulfur compounds. Polyphenols. This is the first phytochemical that I want to discuss because this is the most widely studied phytochemicals of all. It is polyphenols that gives plants their color. So when you see the blackberry or blueberry or raspberry bright in colors, it's probably because of the polyphenols that they have in them. I was discussing about the phytochemicals in plants, and then we were starting to discuss about polyphenols. And like I said, polyphenols is what gives plants its color, and then it can help plants protect themselves from the harmful effects of ultraviolet radiations, and then its aggression. If there is an attack by a pathogen, it could be protected because of the presence of polyphenols. So polyphenols could be classified into various groups based on the number of phenolic rings that they contain, and on the basis of the structural elements that bind these phenolic rings to one another. The different categories of compounds that come under polyphenols will include phenolic acids, flavonoids, stilbenes, lignans. Polyphenols have been scientifically proven to have cardioprotective effect, anti-cancer effect, anti-diabetic effect, antioxidant effect, and neuroprotective effects. So, for example, some of the polyphenols that are well known are quercetin, resveratrol, camphorol, caffeic acid, myrcetine. These are all polyphenols which are very well known. Alkaloids. The next category of phytochemicals that I want to discuss about is alkaloids. Alkaloids are these bitter tasting phytochemicals. So the bitter gourd, when you eat that and it tastes really bitter, it is because of the presence of alkaloids. Now, why do plants have alkaloids in them? So that they could protect themselves from herbivorous organisms that would like to eat them. The alkaloids are mainly characterized by the nitrogen atoms that are present in their structures. And you could classify alkaloids based on the structure, like quinolenols, isoquinolines, pyrrolidins, pyridins, terpenoids, steroids, all based on structures. We could also classify alkaloids based on the plant groups in which they occur. One of the very first alkaloids which was isolated was morphine. It was isolated from opium poppy as early as 1804. Alkaloids are scientifically proven to have antibacterial activity, anti-inflammatory, analgesic. And they've also been studies that say that they can have local anesthetic effect, anti-tumor activity. All these have been attributed to alkaloids. Examples of alkaloids would include, obviously, morphine, quinine, atropine, caffeine, nicotine. The next category of phytochemicals that are present in plants are terpenoids. Another name for terpenoids are isoprenoids. So isoprenoids or terpenoids, these are aromatic compounds, and these are among all the phytochemicals that we see in plants. Terpenoids are probably the most diverse ones, and they have various medicinal properties, and they can help the plant protect themselves from abiotic and biotic stresses. And they're characterized mainly by this structure that is shown here. This is called the isoprene unit. An isoprene unit is a 5-carbon structure, and this is found in various combinations in the different types of terpenoids. So according to the presence of the carbon atoms, we could classify terpenoids as monoterpenes, diterpenes, sesquiterpenes, etc. They have antioxidant effects, antihypertensive, anti-cancer, anti-malarial effects. Examples of terpenoids will include artemisinin, taxol, menthol, geraniol are all examples of terpenoids. Phytosterols, another category of phytochemicals are phytosterols. Phytosterols, I would say, is analogous to cholesterol. So it's the plant version of the cholesterol that we find in animals. In animals, the purpose of cholesterol is mainly in the plasma membrane to help with its fluidity. The same way, phytosterols are present in the cell membrane of plants. So phytosterols are of two types, sterols and stanols. Sterols, if you see, they have a double bond and therefore they're unsaturated. And stanols, if you see, they have no double bond and therefore they are saturated. Phytosterols, they have the ability to compete with cholesterol in absorption. Therefore, a diet rich in phytosterols can lower serum total and LDL cholesterol concentration. Phytosterols are anti-inflammatory, and they have proven to induce apoptosis in cancer cells. Examples of phytosterols will include sitosterol, camptosterol, stigmasterol, brassicatosterol are all examples of phytosterols. And the next category is organosulfur compounds. Like the name implies, these are compounds which are sulfur containing. They have sulfur in them. So they have sulfur containing organosulfur compounds can be basically found in vegetables like broccoli, Brussels sprouts, cauliflower, onion, then garlic. All of them have this organosulfur compound. So if you taste of these vegetables, they have this specific taste, and then they have this really strong flavor. So all that is probably because of the presence of the organosulfur compounds in them. Organosulfur compounds have been proven to have anti-cancer activity, antifungal activity, cardiovascular, anti-cancer, antimicrobial activities. And they're classified basically based on the functional groups to which the sulfur is actually attached. So they are so some of the categories of organosulfur compounds are thiosulfide, sulfones, thioesterate, sulfoxame. So these are all categories of organosulfur compounds. Examples of organosulfur compounds include allicin, thiophenes. So having said so much about the different types of phytochemicals present in plants, the next part of the talk is to discuss about what methods do we have to research on these phytochemical compounds. So there are so many medicinal plants and a lot of them are unexplored. So if you happen to come across a medicinal plant that is unexplored, and if you want to start beginning to analyze the phytochemicals present in them, the very first step would be to start with phytochemical stream. It is during this step that we would try to figure out what kind of phytochemicals are present or what kind of phytochemicals are absent from a certain plant or a certain plant part. This begins with the collection of the plant parts or the plants. The very first step is to collect the plant, identify it, and choose which plant part that we want to work on. And the next step is to start preparing the sample. So then we would try to clean it, dry it, powder it. So all that comes under sample preparation. Once the sample has been prepared, it is ready to go for the extraction stage. So this is a very important stage. What solvent we choose and what method we choose to extract plays a huge role on what kind of phytochemicals we would ultimately end up isolating. Simple methods of extracting the plant compounds include powdering the plants finely or coarsely or homogenizing them and allowing this to interact with the solvent. The solvent that we choose will depend upon the polarity of the compound that we want to isolate. Supposing we are trying to isolate a polar compound, then the solvent of choice should be a polar one. And if the aim is to isolate something like a non-polar compound, then the solvent of choice should be a non-polar one. Now, most often we don't have the choice to determine what type of compound we're isolating, since we are starting with phytochemical screening, but might not even know what kind of compounds are present. In that case, the best way to go would be to start for sequential solvent method, where you use multiple solvents on the same plant powder in a sequential order. Usually, we would go from non-polar towards a polar solvent. The most polar solvent that we would probably use is water. So water, ethanol, methanol, chloroform, ether, acetone, all these are different types of solvents that we could use for extraction. And like I said, we can choose them in the order of polarity. Soxalate apparatus is a widely used one for phytochemical screening. It's like a mini distillation unit, if I should say that. So it has a little bulb at the bottom where we put the solvent, and then we allow the solvent to boil. The solvent boils, evaporates, and then there is this little mechanism to condense the solvent. And then the condensate of the solvent falls into our sample. And then it's about the sample getting continuously flooded with the solvent, and then the solvent ultimately gets collected in the bulb at the bottom, and then at one point in time you can collect the solvent which is rich in the extract, evaporate the solvent, and take out the plant compound. So if you observe, the main principle behind extraction is to allow the plant sample to interact with the solvent so that the active ingredients could be pulled out of the plant parts. Using soxalate or conventional methods of extraction, the problem is that they use a lot of solvent, and then while evaporating, you are sending it out the vapors into the atmosphere, and then that can create environmental hazards. A lot of energy is required. To minimize all these things, some newer techniques have evolved. One is called microwave assisted extraction. This technique uses microwave to extract compounds. So we would basically mix the solvent and sample, and then subject it to microwave, maybe low or medium microwave. And then the time of exposure will depend on what type of sample you're trying, what type of compound you're trying to extract. However, this technique takes only very less time and very less energy as compared to the conventional methods. Ultrasonic assisted extraction is also another technique where instead of microwave, we would use ultrasonic waves. 20 kilohertz or about that is ideal to break the cell open, and then the contents could be released. Supercritical fluid extraction is also another technique, but then you have to have some sophisticated instrumentation to have supercritical fluid extraction. Here, the solvent used is supercritical fluids. Supercritical fluids can exist as both in the gaseous state and in the liquid state above a critical temperature and critical pressure. The most commonly used supercritical fluids are carbon dioxide, which can exist as both liquid and solid beyond a certain temperature and certain pressure. Again, allow the sample to interact with the supercritical fluid, and then an extractor allows us to extract permits the interaction, and then we can recover the solvent and extract. Phytosonic extraction uses phytosols. Phytosols are non-chlorinated fluorohydrocarbons, and we can use this also for extraction. Again, these are all less energy intensive and more eco-friendly. After the extraction is done, we can proceed. We have the extract in hand, and then the next step is to proceed with phytochemical screening. So preliminary screening can be done with various biochemical tests. Tests for alkaloids, phenols, saponins, tannins, flavonoids. A lot of phytochemicals tests are available. These tests have been standardized by various scientists, and we can use these tests to do a preliminary screening of the extracts that we have. most commonly used tests are have been proposed by Harbon, Treason-Evan, Sofovora. So we can use the test that they have proposed to do a preliminary phytochemical screening, which will give us an idea of the presence or the absence of the phytochemicals. Having carried out phytochemical screening, we can move on to more sophisticated methods of analysis using some techniques, and I would like to throw some light upon these techniques in this section. Thin layer chromatography has been used for many years for phytochemical analysis. So this is a technique that allows us to separate the components in a short period of time. So thin layer chromatography, as the name implies, it uses a thin layer of silica or alumina, which is coated on some solid substrate. We would spot the extract on a thin layer, and then the plate is allowed to interact with the mobile phase. Separation of the compound from your extract will depend upon the affinity of the compound with the mobile phase or with the stationary phase. Now, the mobile phase is a mixture of solvents. And then in this picture, if you see the mobile phase is in blue, and then the TLC plate itself is a little rectangle, and you can see the spots right here. So when the spots of the extracts that we have placed on the plate interacts with the mobile phase, it is carried on the plate. However, the speed at which the different components in the spot will move will depend upon its affinity. So a compound which has a high affinity for the mobile phase will move at a faster rate along with the mobile phase, while the compounds that have a high affinity for the stationary phase, which is the silica coated on the plate, will stay back and then migrate slowly. So by this method, you're able to separate the various components in the mixture. So once the separation has been achieved, we can scrape out the spots. We can scrape out the spots from the plate and then take them for further analysis. Or directly, if you want to visualize how many different components were present in your extract, we can spray a chemical over it, which reacts to these spots, they give a color and see it. We can sometimes also view the plate under ultraviolet rays. The band that is scraped could be analyzed further by using ultraviolet visible spectroscopy. And during this method, what's happened is the individual components that we have separated from the plate, they are allowed to be absorbed by different wavelengths in the ultraviolet visible spectrum of the electromagnetic radiation. So each component has an absorption maxima at a particular wavelength, and this could be exploited to identify different compounds. Sometimes in the thin layer chromatography process, we can spot standard compound along with the unknown mixture and then try to see if the migration of a certain compound is the same as that of the standard, and that also helps us to identify the components. A more sophisticated chromatographic method is column chromatography. In column chromatography, what we do is instead of coating the material, the stationary phase on something, we pack the stationary phase in a column, just like it is shown here. So the column is usually packed with material like silica or alumina or starch, calcium carbonate. All this can be used as a stationary phase. And then we pack the column without any cracks, and then the sample or our extract is mixed with a mobile phase, a solvent or a mixture of solvents, and then it is layered on top of the column. And the mobile phase is allowed to flow through the column. The principle here again is the same. Your extract has various components. Each component is going to move through the column depending upon its affinity towards the mobile phase or its stationary phase. Like in this example, we see that the green colored compound moved fastest, probably because it had the most affinity towards the mobile phase. So any compound that has a higher affinity for the mobile phase will move faster through the column. The ones which have A lower affinity for the mobile phase, or the ones that have a higher affinity for the stationary phase, will stay back in the column. So from this example that is shown here in this picture, I could probably say the blue colored one stayed back because it had a higher affinity for the stationary phase. The green colored one eluted first because it had a high affinity for the mobile phase. The fractions or the eluents are collected one after the another in the order in which they are allotted, and each fraction could be further analyzed and tested for their purity, or they could be further analyzed for the presence of phytochemicals in them. HPLC is a very efficient technique to analyze the elements that we have from the column. HPLC, which is expanded as high performance liquid chromatography, is a very efficient technique to help us analyze the various components that are present elevate from a column chromatography. Basically, in HPLC, the sample along with the mobile phase is forced through a column. Now, the speciality of a column in HPLC is that unlike a column in column chromatography, it's much thinner and it has smaller particles, it's tightly packed, and it is able to withstand high pressures. So we send in the sample along with the mobile phase under very high pressure through a column in HPLC. It's a very sophisticated instrumentation. And again, the principle is still the same. The separation is still based upon the differences in the affinities of the different components that pass through the column. So a detector will detect the separated components and send the information to a recorder. And the recorder gives us a chromatogram based upon the retention. Very efficient quantitative technique as well. Apart from telling us if our compound is pure, this technique can also tell us the amount of the particular compound. So the area or the height of the peak gives us the concentration of the certain compound. It's a very sensitive technique. Two different versions of HPLC exist. One is called the normal phase HPLC, and the other one is reverse phase HPLC. A normal phase HPLC, here the stationary phase is polar, while in reverse phase HPLC, the stationary phase is non-polar. So we would choose it based upon what kind of a compound we are trying to take out. UPLC, I would say, is an advanced version of HPLC, uses much more pressure than HPLC. And so the particle size in the stationary phase is, again, much more smaller than what we have in HPLC, uses less solvent, faster analysis time, high resolution, higher sensitivity, or some of the advantages that come with UPLC could be used with various types of detectors. One other widely used technique in the analysis of phytochemicals is GCMS, gas chromatography, coupled with mass spectrometry. So gas chromatography is usually used to separate volatile compounds. So we would vaporize the sample solution and pass it through a chromatographic column. And then the mobile phase here is not liquid, it is gas. The gas that we choose is an inert gas. So we pass a vaporized sample through a column, and then instead of using a solvent as a liquid phase, like we have seen so far, we would be using inert gas as a mobile phase. Principle is still the same. Components are going to separate based on their difference in affinity towards the stationary and the mobile phase. Therefore, depending upon their affinity, they would have different retention times. And when the compounds come out of gas chromatography, they would be sent to a mass spectrum. So that is the speciality of combining GC with MS. So when you combine that, the element, whatever comes out as GC, they're going to be sent to another instrumentation where the ionization of whatever comes out takes place. And then these ions are accelerated, deflected, and the ions are detected using a charge to mass ratio. And the mass spectrum which is obtained is compared usually with existing database which has the mass spectrum from known compounds. So this is a widely used technique. If you see research articles on phytochemical analysis of medicinal plant compounds, and there are so many reports which would say that the results from the GCMS was compared with NASD database, and then severe magnetic resonance spectroscopy, or simply most commonly known as NMR. And so NMR is used to identify the carbon hydrogen framework, mostly of organic compounds. So NMR relies upon the principle that the nuclei of certain atoms could act as magnets, and then these magnets have two different energy states depending on whether they are aligned with an external magnetic pole or whether they are not aligned with an external magnetic pole. So within the magnetic field, we can allow the nucleus to go from one energy state to another energy state by supplying radio frequency waves. So when radio frequency generator is used in this instrumentation, this would irradiate the sample with a short pulse of radiation, and this radiation will allow to cause the shift, or let's say it causes the resonance. So a detector can measure the energy that is released during the nuclei's return to a lower energy state, and then we obtain a spectrum that basically provides us with a chemical shift. So this indicates the chemical shift basically indicates the molecular environment of the nuclei that is present. So we can have proton NMR or we can have C13 NMR depending on what type of nuclei we are trying to study. Structure determination can also be performed using infrared spectroscopy. During this technique, the sample is exposed to infrared radiation, or IR, simply just to say, and then the atoms are allowed to absorb the IR. So the thing with IR is that not all type of bonds are capable of absorbing IR. So a bond that is polar probably would be able to absorb IR better than one that is non-polar. So different functional groups can absorb IR at different wave numbers, and the IR spectrum basically gives us the percent transmittance versus wave number. Some of the medicinal plants that we have worked upon in our labs are Ipomea batatas, Arslake tagala, Calotropis procera, Lantana chimera, and Isomilus malabarica. So from all these plants, we were able to identify the presence of phytochemical compound and elucidate the structure of some compounds as well. From Ipomea batatas, using TLC, column chromatography, HPLC, and NMR, we were able to isolate fluoroipomianol and deduce its structure. Fluoroipomianol is an anti-cancer agent, very potent anti-cancer agent against lung cancer. Arslake tagala was another medicinal plant that we worked upon. Preliminary screening was done, UV visible spectrophotometry, TLC, GCMS were all used to identify various phytochemicals from Arslake tagala. Some of the compounds identified in A tagala were ethanol, congol, ginepine, capric acid, lauric acid, and preliminary screening revealed the presence of alkaloids, phenols, and flavonoids. Calotropis procera. In this particular plant, we were able to identify various polyphenols using UPLC. Lantana chimera, volatile compounds were identified using GCMS. And Isomilus malabarica, preliminary screening revealed the presence of some basic compounds like phenols, proteins, carbohydrates. Having said so much about phytochemicals, the way to identify them, the different methods available to elucidate their structure, the next question is should we actually rely upon plants which are growing in their natural habitat to isolate all the phytochemicals? If all scientists were to do that, if mankind were to rely completely on naturally available plants and their parts for isolation of phytochemicals and for their medicinal needs, then we would end up destroying the biodiversity of this universe. So that is when plant tissue culture comes into the picture and helps us protect the biodiversity. A very efficient method to generate the biomass that we need for the isolation of phytochemicals would be to rely upon cell suspension culture. This, I would say, is a strategic approach for the large scale production of phytochemicals, and at the same time, it would help preserve the biodiversity. During cell suspension culture, we culture plant cells in liquid nutrient medium. And then during this technique, we aim to produce a suspension of single cells. Now, for this procedure, we have to first standardize the production of callus. So it starts with the production of an organized mass of cells called the callus. So we have to standardize the procedure for the production of callus. Callus could be produced from any plant part. We can have calli starting from root explants, stem explants, or internodes, nodes, leaves. So from any part of the plant, we can standardize a procedure for the production of callus in vitro. From the callus, the cells in the callus could be allowed to dislodge using a shaker. And then when you have a single cell suspension, this could be used to seed a larger culture. Optimization of the culture medium is one thing that we have to always do with cell suspension culture. And then if you're going in for large scale cell suspension culture, then we have to choose an appropriate bioreactor system for the purpose. Cell suspension culture can be carried out in batch mode or in continuous mode. In a batch mode, what we would do is we would heat the medium, set the pH, aerate, agitation, everything we set to a certain point, and then don't disturb the culture for a certain period of time, and then harvest whatever we want at the end of it. So there is no way we are inputting a fresh medium in this culture. It's kind of like a closed culture. However, in a continuous culture, we allow the input of fresh medium and the harvesting of spent medium. So the mode that we want to choose will depend upon what kind of phytochemical we are looking at. Is the phytochemical present inside the cells, or is it released outside the cells? So depending on that, depending on whatever is available in our laboratory or what we are looking at, we will have to choose the different types of biocultures that we have to use. The bottom line is that cell suspension culture can give us a huge amount of biomass that we can work upon. When we are working upon Ipomia batatas, so Ipomia batatas, the common name for that is sweet potato. Sweet potato, we know that it's a root tuber. If we have to isolate phytochemicals from something like a root tuber, harvesting it from natural environment obviously is not the right way to go, because it will destroy the plant. And then even for a small amount of phytochemical, I would have to have a lot of sweet potatoes, for which a large amount of land, which could be otherwise used for other purposes, will have to be utilized for this. So that is the reason why we had to go in for cell suspension culture when we wanted to isolate the anti-cancer compound from sweet potato. So we initiated a cell suspension culture. We went in for the batch mode. We used a stir tank bioreactor for that. And then by the end of the batch mode, we obtained a lot of cells, which we could use for the isolation of Ipomia and all. Another advantage that comes along with the cell suspension culture, in addition to whatever I mentioned so far, is the amenability for elicitation. Cell suspension culture allows us the opportunity to induce the cultures to, produce various secondary metabolites. what mechanism stimulates a plant to produce secondary metabolites it will depend from plant to plant, and where the secondary metabolite is concentrated is also can vary from different parts of the plant to different parts. So elicitation is a method that induces the production of secondary metabolites. For instance, when we were trying to make 4-ipomenol, 4-ipomenol could be elicited by a fungal, by the presence of a fungi or a fungal component in the medium. So we had to use the fungal component in the medium to elicit the production of 4-ipomenol. So this is possible in cell suspension culture. We can use some kind of an inducer to enhance the production of the secondary metabolite. Now, an important area of medicinal plant research is bioactivity studies. The phytochemicals isolated in their pure form or the crude extracts are studied for bioactivities. Bioactivities will include antimicrobial activity, antibacterial activity, antiviral, antifungal, anti-carcinogenic activity, anti-inflammatory activity, antioxidant activities. All these fall into the category of bioactivity studies. Some of the most commonly performed bioactivity studies will include antibacterial activity. So antibacterial activities are usually assayed by agar well diffusion method. This is a technique where we have an agar plate, and then the microorganism is cultured on the plate by a swab, and then we make wells on the agar plate. The plant extract at various concentration is put into different wells, and after incubation, if the plant extract has the potential to inhibit the growth of the microorganism, then it would have a zone of inhibition around it. Larger the zone, the higher is the efficiency of the plant to inhibit the activity. Kirby-Bear disk diffusion method is another method to test the antibacterial activity. In this method, what happens is that we, instead of using wells, we basically use filter paper discs, which are dipped in the plant extracts, and then still measure the zone of inhibition. Broad microdilution method is a standardized procedure which can help us measure the minimum inhibitory concentration or the minimum bactericidal concentration of a plant extract. In this technique, what we do is we use well plates to culture the microorganisms along with the plant extracts. And the minimum inhibitory concentration is the concentration of the plant extract at which you see no visible growth of the microorganism. While the minimum bactericidal concentration is the concentration at which the bacteria are killed, basically. So if you take out, at that particular concentration, if you take out the microorganism, the culture mixed with the extract, and then if you subculture it on a fresh medium, you should see no growth of the microorganism at all. Time kill kinetic assay is another technique that allows us to assay the bactericidal activity or the bacteriostatic activity of the plant extract. Usually after determination of the minimum inhibitory concentration, we would take that concentration, and around that concentration, we will work around to see the time kill kinetics assay. A biofilm inhibition assay is a technique to figure out if the plant extract has the capability to inhibit the formation of biofilm. So the first step is to actually allow the formation of the biofilm. In a well plate, we would allow the biofilm to form by incubating it. A staining technique can reveal the formation of the biofilm. And then the plant extract could be allowed to interact with the microorganism, and its effect on the stabilization or the destabilization of the biofilm could be studied later on. Antifungal activity measures the potential of the plant extract to inhibit the growth of a fungus. Broad dilution method and time kill kinetics assay are still used with antifungal activity. Anti-HIV activity is something that falls under bioactivity studies, because we know that HIV is one virus which is posing a huge challenge to humankind. So combating the virus is not easy because it develops drug resistance very fastly for a lot of reasons. It's combating HIV is becoming hard. So plant-based medicine is the alternative for that. So in order to determine the effect of some medicinal plants, we had used Cleistanthus colinus and Aristolochia elegans in the past in our labs to see if they can have an effect on the growth of HIV in culture. So there are various methods available to assess anti-HIV activity of plant extracts, but I'm just going to discuss the one that we used in our laboratory. So what we basically did was we collected lymphocytes from HIV positive blood. So we used HIV positive blood, isolated lymphocytes from that, and cultured these cells in incubator. And then we added different dilutions of the plant extracts, allowed it to incubate again. And the method that we used to figure out how much of the virus was active was by using P24 antigen expression. For this, we used a commercial kit, and then we assayed the expression of P24 antigen, and that gave us an idea of how much the extract was able to inhibit the anti-HIV activity. In both the extracts, we found that they were able to inhibit HIV in a dose-dependent manner. Cytotoxicity assay is performed to evaluate plant extracts efficiency to inhibit cells, inhibit the growth of cells. So this gives us an idea about how efficient a plant extracts are capable of, inhibiting the growth of cells in culture. One commonly used assay is MTT assay. It relies on the principle that if a cell is metabolically active, then it is able to reduce MTT to formazon, and then we can measure. Formazon is colored, and then we can use spectrophotometric techniques to measure how much this activity was present there. Antioxidant activities are widely studied. So many plant extracts are being studied today for their antioxidant activities. A lot of labs are carrying out their research in this area. In general, the methods for assessing the antioxidant activity of plant extracts could be either assays associated with their capability for radical scavenging, or assays associated with their capability to inhibit lipid peroxidation. DPPH, radical scavenging assay, nitric oxide radical inhibition assay, hydroxyl radical scavenging, ABTS, radical scavenging assay. These are all assays that fall into the category of assays associated with radical scavenging. And then TBARS assay and inhibition of beta-carotene bleaching. These two help us to fall into the category of lipid peroxidation assays. So the various medicinal plants that I and my collaborators were able to work on so far have been Aristolochia tagala, Aristolochia elegans, Calotropis procera, Cleistanthus colinus, Lantana camera, and Isomilus meliberica, Ipomea batatus, and Eagle marmelos. So these are the different medicinal plants on which we were able to do phytochemical analysis, isolation of bioactive compounds, and various bioactivity studies. So in conclusion, I would like to say that research on medicinal plants, it opens a lot of opportunity for the discovery of novel lead compounds. Out of the 250,000 medicinal plants that have been discovered so far, only 10% has been analyzed for the presence of bioactive compounds. There are so many plants which are unexplored yet. In these plants lies huge potential. Some of these plants might be harboring very potent antiviral compound in them, which we need at this point of time. And then some of these plants probably have a potent anti-cancer drug in them. Some of them probably have the lead compound for inhibiting the growth of HIV. So it's a lot of potential that is available in medicinal plants. And it is our collective responsibility to protect this biodiversity and pass on the treasure for generations to come. Having said this much, I would like to thank you all for your patient listening. And these are the next part of this thing is all about references that I had used for this. Thank you so much for patient listening. And if you have any questions, we can ask now.
Thank you, ma'am, for an informative session and for sharing your knowledge on various phytochemicals, their screening, techniques for analyzing them, as well as the bioactivity studies. We have a few questions here, ma'am. So shall we get into the Q&A session?
Of course, of course.
Okay, ma'am. The first question here is what is the peak value that is expected from UPLC when comparing to HPLC for any phytochemicals extracted from any medicinal plants? And if it is good, what may the resolution of the purity in UPLC be?
Okay, resolution in UPLC is definitely much higher than what you would have in HPLC. The peak value, if you're operating under the same with the same solvent, so the retention time should ideally for a certain compound, the retention time is very specific, and it will depend on what kind of compound we are trying to extract, what solvent system we are using, and what column we are using.
Thank you, ma'am. The next question is how to choose the solvent for column chromatography.
So supposing your compound is nonpolar and your solvent is also nonpolar, then it is likely to move very fast with the solvent, right? So you should, based upon the polarity of what we are trying to isolate, we should choose a solvent system. Solvent system is usually, instead of going for one particular solvent, we will usually try to mix the solvents so that we are able to separate a wide variety of compounds.
Thank you, ma'am. The next question is, NMR is the most used technique for structure determination of phytochemical novel compounds rather than MS. Why is it so?
Why do we use NMR more than MS? Okay. See, both of them, I would say have their own limitations and advantages. So even when we were trying to elucidate the structure of the compound, we had to use both MS and NMR for structural comparison. Really, I would say that a combination of both would be the right practice to go for to elucidate the structure.
Okay, ma'am. The next question is, what are the most essential analytical tests to confirm the medicinal ability of a particular plant? And could you please elaborate?
What tests do we have to figure out the medicinal ability of the particular plant? I would say that going in for bioactivity studies is the best way to confirm the medicinal ability of a certain plant. So if we can antimicrobial tests ascertain whether the plants have any activity against a microorganism, right? And then when you go for cytotoxic assay, you're able to see if it can prevent the growth of cancer cell lines. And when we go on to study about anti-inflammatory activity or antioxidant activity, that gives us an idea about the medicinal potential of the plant as well. So biofilm inhibition study was also discussed in today's presentation. So that is also another way of ascertaining a plant's medicinal activity. So medicinal activity is usually ascertained by performing various bioassays, and there are bioassays can also be performed in vivo in animal models. The only thing is that obtaining ethical committee approval and working on animals, we have to have permissions and everything for that. But if you have that, that is also the next level for ascertaining the medicinal value of a certain plant.
Thank you, ma'am. Naturally, which phytochemical present in maximum amount in the known order? And if you have any idea for new plant species if identified?
Let me understand. Is the question asking about what is the order of the phytochemicals? No.
I'm sorry, ma'am. Yeah, I'll try rephrasing that. Naturally, which phytochemical is present in maximum? And if you have any idea which new plant species is identified from it?
Okay, which phytochemical present the most demand? Um, okay, all different plants have different phytochemicals in different amounts. Okay, so it would be hard to say which is the most amount of phytochemicals, but we cannot. I don't know how if I can say. So some plants have a lot of polyphenols. Some of them have a lot of terpenes, and depending upon the plant. But phytochemicals, I would say, is widely studied. They are present, like whatever plants I analyzed so far, all of them had a good amount of polyphenols. So that is one thing, just in case if that answers the question. And for new plants identification, say that are all most of the 10% of the plants alone have been explored, and then we have a lot of plants still explored. So at this point of time, I'm sorry, I'm not able to give one particular plant that is not because there are so many plants, and then some portions of it is explored, some is not explored. In a certain plant, the root has been studied, the bark has not been. In some plants, the fruits have been studied, the leaves have not been studied, right? So even in one plant, in different parts have been studied. In some plants, polyphenols have been isolated, but then nobody studied the alkaloids. So then there's a lot of variation. So yeah, so we cannot actually say that if there's any new plant, it's left for the scientists to discover that. Yeah.
Yeah, okay, ma'am. The next question is it possible to identify a novel compound using any of the techniques that you've mentioned?
Yes, why not? It is possible to identify a novel compound using this technique. See, supposing you start with column chromatography, right? And then you isolate a fraction, and then but then you do not know what that is. That probably might contain a novel compound, and a lot of novel compounds are discovered like this actually. So in all these techniques, we are trying to isolate the compound in a pure form. Now, whatever fraction we have to know whether it is pure or not, that is when we go in for HPLC, right? So then a single peak in HPLC would tell us if it's pure or not. So once you have a compound in a pure form, once you're able to take it out in a pure form by refining your procedure for isolation, if you're able to take out a compound in a pure form, then structure elucidation by NMR, IR, MR, MS should give you an idea for figuring out the structure, and then that you could probably get a novel compound. So many novel compounds are getting reported like this. Yeah.
Thank you, ma'am. The next question is, it's related to the previous question. How can one go for advanced technique to detect novel compounds from plants, and which would be the most cost-effective method to do that?
So an advanced, how should one go for an advanced technique to detect the phytochemicals? So we start from preliminary screening, and then we slowly proceed, towards various techniques. I would suggest that you start with isolating the compound in a pure form. So you have to standardize the procedure in a simple way to isolate it in the pure form. Just try to verify the purity of it using HPLC, and then for the structure elucidation. The best way to do is to go and find a lab which already has an instrument, and then try to collaborate with them and see if they can figure out the structure for us. So it's not possible that every lab has all the sophisticated instruments to do it. So the best way to go would be to collaborate with other scientists who are working on it, and they should be able to work around and tell you the structure. That's the cost-effective ways. Otherwise, investing on instruments, I wouldn't say it's a cost-effective way. Yeah.
Okay, ma'am. Thank you, ma'am. The next question is, why is callus suspension culture production very sensitive when compared to microbial or cell culture techniques?
Okay, choice of aeration, agitation always is a choice when always is a challenge when culturing plant cells. When we were working on cell suspension culture of hypoamebitatis, this was a huge challenge that we faced. So we had to standardizing the aeration and agitation was a huge challenge, and then we wanted to use a stir tank bioreactor, but then we want to make sure that the cells are not disrupted because of the agitation speeds. We had to have an optimal aeration as well. So standardizing that was a huge challenge. So these are the things that pose a challenge while using cell suspension culture. Another thing that comes to cell suspension culture is the establishment of the cell suspension itself. So microbial cultures are much easier to establish, while to standardize the protocol for the production of the callus and then to suspend it in a solution to find out an optimal number of cells that could be seeded into the bioreactor. All that poses a huge challenge. So we have to use specifically designed reactors for plant cells culture, and even that comes with a lot of challenges.
Thank you, ma'am. The next question is there any medicinal plant used for treating genetic disorders?
See, some of the phytochemicals have been proven to have protective effects on neurons. So some genetic disorders are neurological. So then if you want to connect it that way, yes. So there are a lot of medicinal plants which can help cure various disorders. Or they have the phytochemicals which have the potential to combat various genetic disorders. But however, medicinal plants traditionally, and they used it, we cannot say that people knew that these were genetic disorders. So like I say, some of the medicinal plants are highly effective in reducing inflammation. A lot of inflammation comes with a genetic disorder as well. So you get the connection there, right? Yeah.
Yes, ma'am. Yes, ma'am. The next question is, what is the Siddha medicine or medicinal plant discovered to treat the HIV positive cases? And if so, what is the recovery range while comparing with ART?
I am not actually aware of a Siddha medicine which is proven to treat HIV. I'm sorry I cannot answer this question because I'm not an expert in Siddha medicine.
Yeah, no, ma'am. That's okay, ma'am. That's not a problem. We'll move on to the next question. If the plant compound does not have antioxidant activity, won't it be used as a medicinal compound? If so, what are the other properties test? Mainly, we have to test for different types of diseases as primary test. What?
Okay.
Okay.
So was that the question, or did I stop in between, Swati?
I don't know, ma'am. That's the question, ma'am. Did you understand it?
So if the question is like, if the plant, if I understand correctly, the question is if the plant does not have show any antioxidant properties, are there other properties that the plant can have, right?
Yeah, that could be medicinal. Yes, ma'am.
Yeah. See, a plant extract that does not show antioxidant activity does not necessarily mean that the plant extract itself is not medicinal. It could probably have other medicinal activities as well. And again, many times, if the absence of the antioxidant activity is not because of the plant's problem, it's probably because of the way we extracted it. Probably the method that you use to extract did not extract the compound that is involved in the antioxidant activities. So that could be a reason why we don't see it. But anyways, in spite of all that, if you do not see, if you do a bioactivity study, specifically antioxidant activity, and then you don't see the antioxidant activity, then probably the plant has other activity. You can test it for antimicrobial activity. You can test it for anti-inflammatory activity, anti-diabetic activity. There are so many other bioassays that we can study on the plant and check if it is there. And the key thing in all this is to have the right compound. So we should know what we are trying to extract, and then we should go from there.
Okay, ma'am. Thank you, ma'am. And the last question is, you told that alkaloids such as morphine and nicotine help to protect plants from animals. The person who's asked the question has come across a study that states chocolates contain certain amount of nicotine, which makes people to get addicted. How does this compound help in protection of the plant?
Various alkaloids. Okay, alkaloids, like I said, if you see they have a nitrogen. The alkaloids' main thing is the bitter tasting. That is the key thing in alkaloid. So a plant that is bitter tasting obviously is not preferred by a herbivorous organism, right?
Okay. Thank you, ma'am. With that, we end the Q&A session. I'll thank you once again for taking your time to answer all of these questions, ma'am. Thank you, ma'am.
Thank you, Swatika. I hope I answered the question to the best of my ability. Thank you once again.
Thank you, ma'am. Thank you for a very informative session. Now I call upon Dr. LFA Anandraj sir to deliver the vote of thanks for an informative session. Thank you, ma'am.
Good evening. I consider this occasion as an honor and a privilege to extend my vote of thanks to our guest speaker, Dr. Ramya Mohanraj. Once a great man whispered, feeling thankfulness and not expressing it is like wrapping a present and not giving it. Today, I take the opportunity to express all my gratitude in words. First of all, I would like to propose hearty thanks to our respected chairman, Dr. B. Babu Managaran sir, our managing director, Mrs. S. Jessipriya ma'am, our director, Mr. B. Sasisekhar sir, for giving the opportunity to conduct this international technical webinar on advancement in biotechnology. I would like to express our profound gratitude to our beloved principal, Dr. Vaddi Sheshagiri Rao sir, our dean, Dr. B. Parvavadini ma'am, for providing continuous moral support, encouragement, and guidance. A special mention to our respected HOD, Dr. G. Srikumar sir, for being the catalyst that stimulated us to do the best and standing as a pillar of strength. I extended my special thanks to our guest speaker, Dr. Ramya Mohanraj, faculty, University of Houston, who spared her time in spite of her busiest schedule to grace this occasion. Today, we had a great opportunity to hear your thoughts in the field of medicinal plant, and this will surely encourage us to do research in traditional medicine. She is not only gracious in accepting our invitation, but also supportive in getting other resource person. During our earlier discussion, she is very kind enough to attend our discussion meeting without bothering about the timings. Whenever our organization team requested her to do things to happen without any hesitation, she did to us. Thank you so much, ma'am, for sharing your time and experience with us. We all agreed that your lecture was more interesting and meaningful. An event like this cannot happen overnight. The wheels start rolling weeks ago. I require planning and a bird's eye for detail. We have been fortunate enough to be backed with a team of very proactive and dedicated organizers of this webinar who are all well versed in this job. I would like to thank our third year biotech students who worked behind the scene to make the event happen. I also thank all the participants for the patient listening and maintaining their decorum throughout this meeting. Last but not least, I thank Almighty for gracing the successful completion of this occasion. Thank you.